everyone, welcome to my channel IMAT with Mohan. I am here with another video for past paper of IMAT 2020-2021. I have already covered the biology section of 2021-2022. And as you know, I am just proceeding to cover biology sections of all papers now. So we'll start with biology section of this paper today, and I'll try my best to explain each and every concept clearly today. And if you have any questions and if you have any doubts which are not clear after watching this video, you can always feel free to ask it in the comments. So let's start with questions of biology without wasting any time. Okay, so this is the first question of biology section. So let's start. Which of the following healthy cells contain circular DNA? Circular DNA here means plasmid, right? So this question is really easy, and most of the students know the concept, but many students get it wrong. Like 90% of students get these answers wrong because they just don't understand the complete question. I'll tell you why. See, if you just pause this video, like whenever I explain past paper or something or questions, always pause the video after seeing the question and then try to answer yourself and then watch the answer and explanation. Okay, this is the way how you solve the past papers and all and questions and MCQs. This is how you get. Watch this video just for the explanation and seeing that if you are solving it right or not. Okay, so if this was one advice from me. So let's continue. What are the students just mark one only? Why? Because they know E. coli is a prokaryote, and prokaryotes have circular DNA in their cells called plasmids, and they don't even think of any other option because they know these both are eukaryotes, right? So they just go for one only, which is wrong. Why? Because the students just forget the fact that these both cells have mitochondria, right? Eukaryotes have mitochondria in their cells, and mitochondria have plasmid. Even and if it's like a plant cell or something, it has chlorophyll as well. Chlorophyll also has plasmid, right? Students know this thing that like mitochondria has plasmid and chlorophyll has plasmid, but they just put zero else in this question. And the question says healthy cell. It doesn't say nucleus. These both have a linear DNA or chromosome in their nucleus, but if you consider the whole cell, you have to consider organelles as well, which have plasmids, like mitochondria has plasmid. So, according to that, the option should be 1, 2, and 3, not this one, right? If it says nucleus or something, then you don't, you can just consider like they don't even have nucleus, but still, like if it says nucleus, then you'll think the other way. But for now, just like you have to consider whole cells. This is how the examiner confused students in IMAT. Okay, so you should be used to of these type of questions. Let's move next. Next question is the structure of amino acid asparagine is shown. In each image, V to Z, a part of structure is enclosed in a line. Okay, let's read the question. Which line completely encloses the part of molecule that is common in all naturally occurring amino acids and, and the R group of amino acids? Before doing this, let's just draw a structure of amino acid, which most of you may know from high school and previous knowledge, right? But still, let's draw it. For amino acid, there is a carbon in between, right? On one side of this carbon is COOH. There is always COOH. Okay. On one side, there is an NH2 group. Okay. On the other side, there is a H group. Hydrogen. There is always not a group, a single hydrogen atom. And on the fourth side, there is a R group. And this R group is always different in amino acid, and this is a group which we use to like identify the amino acid, and rest are always the same no matter which amino acid it is. Okay, you all may know this thing, like, but still just to remind it. Okay, in these diagrams, it is the same structure. But what the examiner did is just opened the bonds, like opened the oxygen and hydrogen just to confuse, because most of the students couldn't like understand this way, like this structure of amino acid. This is like uh, it's just an open thing of this, which you all know. You are familiar with this, right? So he's just asking to like mm, uh, to identify the molecules which are same in all amino acids, which is this, except for R group. The whole thing is same in every amino acid and the R group. Okay, so we just need to identify that, which is easy if you can identify the middle carbon. For these type of questions, you just need to look for middle carbon, right? If you can find a middle carbon, you can differentiate in everything, because we know that middle carbon is surrounded by these things, and we can separate them, right? So if you see these questions, uh, uh, here middle carbon is this one, having COOH on one side, hydrogen, NH2, and this whole group will be the R group. All the remaining thing will be R group. And here none of like R group or common thing is separated by the circle. It's like a mixture of something. So this option is right. Oh, this option is wrong. In this one is the R group, and again this option is clearly wrong. In these options, if you see, this carbon is the middle carbon, right? On one side there is NH2, hydrogen, COOH, see, and this complete is R group. Here this line or this circle just separate the whole common group from our group, right? So this is first part of our answer. X, like common group of all amino acid will be X. Only two options have X and X on this side. So we'll just look for these options. One is Z and Y. We'll like we have to see both of them. Okay. In this one, this is middle carbon, right? This is COOH, NH2, and hydrogen, and the remaining one will be R group. This whole thing is R group, which is separated. So, 5 only, row number 5 will be the answer. Okay, so, um, but still, let's see Z as well, just to confirm our answer. Z is in this middle carbon is this one, COOH, NH2, hydrogen, and the remaining whole thing will be the R group. And like it just circles half of it, so it couldn't be right as well. So row number 5, answer number B is our right answer. Okay, so let's move next. Okay, so the next question is, the occurrence of which of the following could allow you to distinguish between processes of DNA replication and transcription? Okay, so all of you must be familiar with both of these processes, DNA replication and transcription. Transcription, in which we make like messenger RNA out of DNA, and DNA replication, in this we just replicate the DNA, like DNA out of DNA. Okay, so uh, like question asks which process allow you to distinguish between them. It means we need to like choose the option which is different in both the things, like which is different between replication and transcription. Okay, so to be more clear, I'll show you another slide or diagram. Yeah, here it is. This is replication and this is transcription going on. So we can just compare our options with this. Okay, first option says breaking of hydrogen bond. So let's see. In replication, we do break our hydrogen bonds between the nucleotide bases from here, right, to like replicate it before. And in transcription as well, we need to break these hydrogen bonds between the nucleotides, same as here, to make a messenger RNA. Okay, so breaking of hydrogen bond couldn't be correct because it's present in both. But the second option is formation of hydrogen bond. Hydrogen bonds are breaking here, and the new in replication you can see the new hydrogen bonds are forming here between new DNA and a previous DNA strand, right? So this is true for this, and in transcription as well, new hydrogen bonds are formed between messenger RNA and DNA strand. In the cases, hydrogen bonds are formed. So this couldn't be correct as well. Second is formation of phosphodiester bond. Where is phosphodiester bond? You all may know that phosphodiester bond is between two nucleotides joined together. In this line, these line are phosphodiester bonds. In these as well, A and U, these both nucleotides are joined here together by phosphodiester bonds. So phosphodiester bonds are formed between both of them as like in both the processes as well. So it means none of these options are different in DNA replication or transcription. So we can't distinguish the processes using these things. So none of these would be the answer. I hope it's clear. So next, let's move to next. Next question says, which of the following will cause the appropriate stimulus of the rod cells in the retina in a healthy human eye? Okay, this is pretty much the theoretical question, like there is no concept in it. So if you have read a human eye or function of human eye or something like that, you will know that there's a rhodopsin in rod cells which breaks into opsin and retinal. So there is nothing to like concepts in this. So I'll just move to the next question. So the next question says that the diagram represents the fluid mosaic model of cell membrane. And if you see the question, it says uh, which one of the following labels 1 to 5 shows hydrophobic region of molecule? So hydrophobic region of cell membrane. So if you know the structure of cell membrane, which most of you may know from high school as well. So you must know that this outer region is hydrophilic, right? In membranes, outer regions are hydrophilic and this middle things are hydrophobic. So the answer is pretty much clear that 3 is hydrophilic, which is here these are lipid tails which are hydrophobic, right? So 3 is the answer. Let's move to next one. Okay, so question number 28 says the diagram shows the animal cells from organism that sexually reproduce. Each diagram shows a single cell contain chromosomes during cell division. Which of the following diagram has a correct description of cells from an organism with haploid number of 4? Assume that sex of the organism is inherited in same way as in humans. Okay, so first note down the things that haploid number is 4. So like some I don't know if it's the case in this question as well, and but in some questions the examiner is confused with this thing because students consider the chromosome number of 4 in a whole question when it says haploid number is 4. If haploid number of chromosome is 4, total number of chromosomes must be 8, right? So you should note this thing as well by reading the question. Okay, so let's move to options. First option says cell from male organisms with a diploid number of 8 during anaphase of first division of meiosis. If diploid number is 8, which we already calculated, if diploid number is 8, then in meiosis phase of 1, if you have studied like mitosis and meiosis even in high school or now if you are studying for IMAT, so you must know that in first division of mitosis, both of like homologous chromosomes are separated. And if the diploid number was 8 in both the sides, we must see like 4 and 4 homologous chromosomes. But in this case, we can see they are not like the homologous chromosome. Actually, chromatids are being separated, and we can see 8 and 8 on both sides. So it shows more to be like mitotic phase rather than meiosis. So option number A is wrong. For the next option B, cell has a diploid number of 8 showing the chromosome during anaphase of mitosis. Again, if the diploid number was 8, in mitosis chromosome numbers doesn't change like chromatids are separated and both sides get 8 and 8 of chromatids or 8 and 8 of chromosomes, right? But in this case we can't see this. It's more like uh, second phase of meiosis, in which the chromosome numbers are already half, and now we are separating sister chromatids, right? So it's like second phase of meiosis, not the anaphase of mitosis. So this option is wrong as well. Let's move to option number C. Cells from a female organisms with 8 chromosomes during metaphase of mitosis. If it's mitosis, in mitosis, homologous chromosomes are separated. Sister chromosomes are separated from each other, so cells do not need to line up like that, right? Cells like the homologous chromosomes line up with each other in meiosis, first phase of meiosis, not in mitosis, not any other process. Like if a line up like these chromosomes are to be separated on one side and these are to be separated on one side, but it doesn't happen in mitosis, right? So this option is wrong as well. Number D, cell of female organism with 4 chromatids during metaphase of mitosis. These are not four chromatids. These are like 4 chromosomes, and each chromosome we can see like 2 chromatids each, right? So it's like 8 total chromatids. So because of this number, it's wrong. And secondly, it doesn't show metaphase as well. In metaphase, chromosomes are arranged like that in linear manner. If it's mitosis, single chromosome, like it will not be two layer, like a single line of chromosome will be arranged just to be separated, like they are about to be separated. So this is not the arrangement as well. Second, let's move to next option. Now, since we are sure that all of the other options are wrong, so this must be correct. But still, we can just see it. Cells from male organisms with 16 chromatids during prophase of meiosis. Prophase of meiosis and 16 chromatids. It has 1, 2, 3, 4, 5, 6, 7, 8 chromosomes. 8 chromosomes and each having 2 chromatids. So it means it has 16 chromatids. It seems to be like a prophase because the nuclear membrane is vanished and we can't see nucleolus as well. So yeah, this option is correct. Just don't confuse between number of chromosomes and mitosis meiosis phases. All the students know this, but because like in IMAT there's a lot of pressure about for timings as well, and these type of questions are meant to waste your time. Like all the students know mitosis and meiosis, but in reading this stuff, the students just panic and go for wrong answers even though they know about these things. So just like consider the timings and these things as well when you solve past papers. Okay. Okay. So the next question is, in 1981, the immature male finch this arrived in the island of Galapagos. And this male finch was similar to finches in the species G. fortis, living on the island. However, G this was 70% larger than this and had distinctly different songs. Okay. Diagram shows how, after observing further generations, a new species has evolved. Okay, let's see diagram. It says that these two crossed and had a new generation, and that F1 generation can cross with the previous the same thing, and like had an F2 generation, and F2 generation F1 and F2 cross and form a new species, a population of a new species. Okay, understand. Let's see the question. The success of new species due to larger bills and body sizes and its unique songs. Which of the following items is or are illustrated by this information? Okay. Number one is sympatric speciation. Okay. Reproductive isolation and post-zygotic isolation. Okay. Firstly, to solve this question, you should know all these terms. You should have read evolution and all. I'll just give the preview of this, but you should know the background of evolution to understand this question completely. This it can't be sympatric speciation. For those who know the definition, may know why. Because according to sympatric speciation, the two species are formed from the single ancestor, like in the same geographical region. Like you can just say that there is no like physical barrier or natural barrier separating the two species from breeding and all. But in this question, it says that these two things can breed or cross with each other. But why these two things weren't breeding was this like because of a physical barrier, the sea. They were living in two different islands. And if there is no physical barrier, they both can breed with each other, right? And form a new species or something. So sympatric speciation can't be applied. Now, now next is the reproductive isolation. Uh, is correct. The reproductive isolation can be applied because reproductive isolation, you can say that two species can breed, like opposite to sympatric, you can say, can breed, but they can't because of some physical barrier is separating them and all. The same thing which the question says. So reproductive isolation can be correct. Next is zygote isolation. Post-zygote isolation. This can't be applied either because this definition says that two species can breed, but they can't form. The new generation can't be formed, or if one generation wouldn't be like formed because of some problems after formation of zygote. Like if like if these two species have this types of isolation, we can't see F1 generation. And if it's so, this thing can't be applied either because we can say that these are breeding and the breeding is successful and new species are formed in the end. So this can't be applied either, right? This can't be applied. This can't be applied. Only this can be applied. So the answer is B, 2 only. The third question, now question number 30 says a single base mutation occurs in a gene. Which of the following is or are possible effects of protein produced as a result of mutation? Okay. Number one option says protein has an altered tertiary structure, which is possible in case of missense mutation. If you have read mutation, you should know these things. Okay. So in missense mutation, what happens is the change in base sequence or codon sequence results in altering the structure of protein, which alters the function as well. Okay, so this situation is possible. Second is protein is short in length. This is also possible in case of nonsense mutation. What happens with nonsense mutation is stop codon is formed between in the middle of the chain due to several mutations or the mutation we are talking about. The stop codon just form in the middle of chain, and because of that stop codon, the protein formation stops where it shouldn't be, right? And due to this, a protein's length is short. So this situation also possible in the mutation called nonsense mutation. Number third is protein is unchanged. This thing is also possible in case of silent mutation. What happens in silent mutation is the codon before mutation and the codon formed after mutation just code for a single amino acid. For a same amino acid, we know that for one amino acid we have like 4 to 5 codes for that. Both of if both of them are just coding for a single amino acid, there is no change in protein structure and neither on the function or something. So three of them are possible. If you have read mutation, you will know these all things. Answer will be 1, 2, and 3, which is B. So okay. Okay, so I'm stopping here at question number 30 because this video is taking too long right now. So I'll make another video for this here part 2 where I'll cover remaining questions. Just see that video for remaining questions. I hope that everything I explained in this video is clear. If there's any doubt or anything which is not clear, just drop it in the comments. I will make sure to answer each of you. So, see you in the next video. Bye.